All right, thanks for coming. This is Leah Riedenbach. She's been the Marine Lab intern. She's going to tell you about our independent research project on macroalgae and nutrients.
All right. Hi, everybody. Thanks for coming. Today I'm going to be talking to you guys about macroalgae and nutrient enrichment in Sanibel, Florida, and about the projects that I've been doing during my Marine Lab internship. So I've been mostly focusing on experiments that determine the relationship between macroalgae and the environment. Specifically, I've been looking at macroalgae and nutrients and macroalgae and seagrass. So macroalgae is a vascular, multicellular marine plant, and there's red algae, brown algae, and green algae, and they're differentiated by their pigment types. Macroalgae is different than land plants because they don't have roots that secure to the ground. Instead, they use holdfasts to attach to things, like rocks, hard surfaces, or sand. They also often become unattached and drift in the ocean in these tumbleweed-like formations, or they get trapped in seagrass or washed up on the shore. Macroalgae is a habitat and food source for many animals, including sea hares, which eat red macroalgae, and they use the pigments that are in the macroalgae as a defense decoy, and they make this purple ink, so whenever a predator tries to eat them, they release the ink, and hopefully it confuses the predator. A lot of people confuse macroalgae for what is called red tide, but macroalgae isn't red tide. Red tide is caused by the dinoflagellate Crania brevis, and they are very harmful to the environment because they release toxins when they die. Macroalgae blooms can also be harmful for the environment, but they're not toxic, and they're mostly just a nuisance to people that want to visit beautiful clean beaches. Macroalgae and seagrass share a similar habitat in this area, which is shallow waters in the bay and off the Gulf of Mexico. They form the basis of the marine food web by converting sunlight and nutrients into organic matter that can be used to feed sea turtles and manatees. They also stabilize the sediment, improve water clarity, and increase the amount of oxygen in the water. There are 7 species of seagrass in Florida, and the most common ones that I've seen while doing research here are turtlegrass, manatee grass, and shoalgrass. Macroalgae often become entangled in seagrass, and part of my research was to determine if this had a negative impact on the seagrass beds by trapping macroalgae and seagrass densities through time. So the water quality in this area is really poor, and that's because before there were no man-made changes to the area, water would flow all the way from the Kissimmee River to the Florida Bay, where it was clean and purified, and there was never a connection between Lake Okeechobee and the Caloosahatchee River. Well, now there is a connection, and the water levels are controlled by a series of locks and dams, and Lake Okeechobee is also connected to dairy farms in the north and sugarcane fields in the south, and that contributes to a lot of the pollution in Lake Okeechobee. And when Lake Okeechobee is released into our waterways, we get a huge dose of those nutrients. And that's causing a lot of the issues that we're seeing today. Local efforts, such as this sign, to reduce nutrients in our waters are good because they raise awareness for the problems that are going around in our water. And this sign is right on Causeway Boulevard. So once you come on the island, that's a sign that all the residents are seeing and tourists. So to determine how nutrients affect macroalgae, I did several lab experiments that allowed me to measure their growth rates in high and low nutrient concentrations. And then I also wanted to focus on one species called Celeria filiformis that's really abundant around here, and I wanted to see how it would perform under different nutrient concentrations and at different timescales. And I used ammonium and nitrate, which are 2 different forms of nitrogen that are found in the water. So I used 4 different species for my experiment: Eucuma isoform, Gardiella sabulata, Gracilaria tigbahi, and Celeria filiformis. As you can see, they all kind of look the same. So sometimes to identify them, you have to take cross-sections of them. Eucuma has a dense bundle of filaments in the center and is surrounded by many small cells. Gardiella and Celeria both have a bunch of loose filaments in the center and are surrounded by a few layers of larger cells. These 3 species are all in the same family, which is why I wanted to compare their growth rates and see how they acted in different types of nutrient, or in high or low nutrient concentrations. Gracilaria tigbahi is the most common species found around here in the Gracilaria family, and that's why I've also picked it for this experiment. As you can see, the cross-section grassal area is a lot different than these other three because there's no filaments in it, and it just has a lot of big, thin-walled cells. So, for the growth experiment, this is where the experiment took place, and I collected algae from stranding events at the Lighthouse and Bowman's Beach, and I cut up the algae into small fragments and put them into either high nutrient water or low nutrient water. And I would change the water every other day to replace any of the nutrients that they took up. And I would measure their fresh weight every week, so I was able to keep track of their growth rate for 8 weeks in a row. The most noticeable difference between the low nutrient treatment and the high nutrient treatment is that the low nutrient treatment lost their pigments after several weeks of the experiment, and the high nutrient treatments kept their pigments for the duration of the entire experiment. So there are two different ways to look at the results from this experiment. You can either compare the different species, or you can compare the low and the high treatments. So this one is comparing different species. Celeria had the highest growth rate of all the species, and Eucheuma had the lowest growth rate of all the species. In fact, in the low nutrient treatment, all the growth rates were negative, meaning that they weren't growing, they were actually decaying. We predicted this occurred because Eucheuma has really thick, tough, solid branches, and they don't respond well to fragmentation. So by day 35, almost all the fragments died. On the other hand, Celeria has really thin branches and fragments really easily, and it can reproduce asexually through fragmentation. So when one fragment breaks off, a new individual can grow from that fragment. This life strategy can lend itself to prolific numbers in certain areas, such as those with high nutrients, and explains why it's the second most abundant macroalgae species in the Gulf of Mexico off the Sanibel coastline. So this is the same graph. I just changed the scale to zoom in and compare the high and low nutrient treatments. Celeria and Gracilaria had significantly higher growth rates in the high nutrient treatment than in the low nutrient treatment. And Agardiella subulata had significantly higher growth rates during the end of the experiment, but not in the beginning. Overall, the growth rates in Celeria and Gracilaria decreased through time. We believe this had to do with self-shading, so the plants grew in these tumbleweed formations, and light couldn't reach the bottom part of the plant, so therefore the growth rate slowed down. Agardiella had a relatively stable growth rate for the entire experiment, and that's probably because there was no self-shading, because they didn't grow in that formation. And this reduction in growth rate is probably due to senescence, which is the process of aging and decaying. Here is probably the decline in the growth rate probably had to do with a combination of senescence and self-shading. So the most important piece of information to take away from this experiment is that macroalgae grow faster in high nutrient water, and some species are more advantageous than others when it comes to high nutrient water. Mark?
What level is high nutrient and what level is low nutrient?
That is a good question. The high nutrient water was around, since I changed the water every other day, it's different throughout the entire experiment, but it was around 30 micromolar. And the low nutrient treatment was just ambient water from Tarpon Bay, so it was around 3 micromolar. So I spiked the high nutrient ones. Okay. So I really want to focus on Celeria filiformis because it seems to be taking advantage of the high nutrient water, and it's very abundant. So that's why I chose to do a nutrient kinetics experiment on it. Nutrient kinetics is also called the Michaelis-Menten kinetics, and it's a formula that can be used to tell you a lot about the physiology of the macroalgae. It can tell you the maximum uptake rate, the nutrient saturation point, and the uptake affinity at low concentrations. To do this, I set up 6 different nutrient concentrations at 0, 5, 10, 20, 30, and 40 micromolar, and I analyzed them for ammonium and nitrate. So ammonium and nitrate were in equal concentrations in each different treatment. During the experiment, I took water samples at 0, 20, 90, and 180 minutes, and I used this formula to determine the uptake rate. And the final graph that you get from this is a graph of the uptake rates versus the concentrations.
So for like 40 micromolar, you had 20 micromolar nitrate and 20 micromolar ammonia?
No, I had 40 micromolar of each.
Oh.
Yeah. So before I go on discussing the results of that experiment, I want to talk to you a little bit more about macroalgae nutrient uptake. Macroalgae need nutrients to grow and to support making proteins and amino acids, and they use nitrogen, phosphorus, carbon, and trace minerals to do this. They also need sunlight and water movement for growth. Water movement reduces the boundary layer between the cell wall and the water, and it allows for greater access to nutrients. They also have a preferred salinity range and a preferred temperature range. When nitrate enters the cell wall, it has to be reduced to ammonium through nitrate reductase activity. When ammonium enters the cell wall, it's immediately converted into amino acids and proteins. That's why, and nitrate reductase activity uses up energy. So most species of macroalgae prefer ammonium over nitrate. So these are the McKellis-Mitten curves that were the results of the experiment. I'm showing here the results at 20 minutes and the results at 180 minutes, or 3 hours. I'm not showing the results of 90 minutes, but I will discuss them further later. These graphs only show the results for ammonium, not for nitrate. I'll discuss nitrate later too. Okay, so the first thing that these curves can tell you about the macroalgae is their maximum uptake rate, which is called the Vmax. During the first 20 minutes of exposure to high nutrients, the Vmax is 252 micromolar per hour, which is pretty high. Higher uptake rates are usually associated with higher growth rates, but that's not exactly what's happening here. Prior to the experiment, I kept all the fragments in low nutrient water, so they were kind of in a nitrogen-starved mode once the experiment started. This first 20 minutes is known as surge uptake. So there's an increase in the uptake rate that occurs when the internal pools of nutrient pools are depleted and they become filled. This is a good life strategy for macroalgae because they're either attached and the environment of the water is changing around them, either from lake releases or natural phenomenon, or they're unattached and they're going through different environments that have varying levels of nutrient availability. So when they have full nutrient pools from, say, a lake release, they can go and they end up in an environment that has low nutrients. They can use their reserve pools until they need to refill them again or until they can't and they'll start to die. So the surge uptake rate represents the rate of the filling of the pools of nutrients and also the rate of amino acid synthesis. The rate lowers at 90 minutes to 78 micromolar per hour and continues to lower even more during 3 hours. So at this time period, the pools are still filling, but not as quickly. And then at 60 minutes, or at 180 minutes, the pools have become full, and this rate represents the rate of conversion of ammonium to amino acids. Okay.
So for these experiments, you're mostly just concerned with nitrogen and not phosphorus?
Correct, because nitrogen is the limiting nutrient in our water for macroalgae. So this phase is called the assimilation uptake phase. Okay, so the Km is the half saturation constant, or the rate, or the concentration at half the maximum rate. This is used to estimate the ability of a species to absorb a nutrient at low concentrations. The Km at 20 minutes is really high compared to some other species that I've seen in scientific literature. A high Km value is associated with organisms that are not efficient at performing uptake in low nutrient concentrations. organisms that have a high Km are often found in eutrophic waters. The Km is a useful measurement for comparing different species, and so a species that performs well in a low nutrient environment would have a low Km. The Vmax over Km is an index of ammonium affinity at low concentrations, and it's also the initial slope of the curve. The affinity is really high during the first 20 minutes during the surge uptake period because the fragments are starved of nutrients. So they really begin to quickly fill their internal pools of nutrients. And they also, at the same time, are converting ammonium into amino acids. The Km, or the Vmax over Km at 90 minutes is about the same. So they're still filling their internal pools, and it decreases at 180 minutes. So their affinity for nitrogen or ammonium is decreased. The R² value is another component of this, and it basically explains how well the data fit the model. And the higher the R² value is, the better the model explains, or the more predicting power the model has. So over time, the R² increases. And this probably occurs because there was a lot of variability in the uptake rate in the beginning because all the fragments had different amounts of nutrients in their internal nutrient pools. Over time, the R² increased, and that probably occurred because all the nutrient pools were filled, and since they're all the same species, they all kind of were converting ammonium into amino acid at the same rate, and they fit the curve better. So the most important things to take from this experiment were that the maximum uptake rate during the first 20 minutes was extremely high, and that means that Celeria can take up to 252 micromolars of concentrations or 252 micromolars per hour in really high concentration waters. The uptake rates were higher during the first 20 minutes than after 3 hours. So pulses of nutrient-rich water are coming from Lake Okeechobee. Celeria can quickly take up these nutrients and continue to grow even after the pulses are over. This means that Celeria takes advantage of the Lake Okeechobee releases or other nutrient additions. So I also used nitrate during this experiment, but the nitrate didn't fit the Macroalgae equation. Because remember, macroalgae prefer ammonium over nitrate. So the uptake rates were reduced. Overall, it had trends of faster rates and higher concentrations, just like with ammonium. And the rates decreased over time. That probably had to do with filling internal pools. This is ammonium, and this is nitrate. So as you can see, the rates of ammonium uptake are a lot higher, especially in higher concentrations. I also did just a long-term 12-hour nutrient uptake experiment to compare the growth rates in high and low concentrations, and it was much faster, 2 to 5 times faster in the high nutrient treatment than in the low nutrient treatment. Alright, so I also did macroalgae and seagrass surveys at 7 sites, all the way from the toll booth to Captiva. When I went to each site, I would take the percent coverage of all the seagrass species present and the percent coverage of the macroalgae present, and at 4 different quadrats per site. To examine the composition of the macroalgae species, I would take the macroalgae from the quadrats, collect them, and identify them to the lowest taxonomic level possible, and I would dry them to obtain their dry weight, because that's the standard way of doing things for macroalgae. With this information, I was able to determine the macroalgae density or diversity through time and space, and also able to examine the relationship between macroalgae and seagrass. So these are all the different locations: Tollbooth, Causeway, Bailey Road, Dixie Beach Boulevard, Tarpon Bay, Wolfert Road, and Andy Rossi. Alright, these are the results for the macroalgae diversity at each site during the 3 different study periods. And I also put on the dominant species that I found at each site and the percentage of the dominant species. Andy Rossi Lane is the furthest site from the Lake Okeechobee releases, and it also had the highest diversity of all the sites, at least during the first 2 study periods. The study periods were December, January, and February. These 4 sites, Andy Rossi Lane, Wolfert Road, Dixie Beach Boulevard, and the Tollbooth, all decreased in macroalgae diversity over the study period. And I also noticed that as the dominant species percentage increased, the diversity decreased. This occurs because some species can take advantage of high nutrient environments better than others, and this exploitation of resources can cause a reduction or elimination of other species. Macroalgae diversity is important because there are many animals in the ocean that depend on macroalgae as a habitat, food source, or both. Humans also use macroalgae as a food source, either directly, as in sushi, or indirectly through a product that is put to thicken ice cream and other products like that. It's called carrageenan, so you might see it on your labels of ice cream sometimes. Dominant species can be detrimental to the habitat because they attach to seagrass or become entangled in seagrass, and they block sunlight, which seagrass need for growth. Some dominant species also have little value as food for humans, and some of them are quite a nuisance. There's one called Hinxia, and the common name for it is called snot algae, and it's pretty disgusting to swim with, and it's very unpleasant. So these 3 sites increased in biodiversity throughout the study period. And those sites are here, here, and the Causeway, which is here. Although the percentage of the species didn't really decrease a whole lot, the diversity still went up. Okay, these sites, Andy Rossi Lane, Tarpon Bay, and the Causeway, at the beginning of the surveys, they were all dominated by drift macroalgae. By the end, they had all changed compositions to a filamentous algae that was attached to seagrass blades. By the end of the study, all these sites were over 70% polysiphonia. In addition, Dixie Beach Boulevard and Bailey Road, which is really close to it, were dominated by the filamentous algae during the whole study period. Dixie Beach was dominated by polysiphonia, and Bailey Road was dominated by Hinxia, the snot algae. Overall, there was a trend of decreasing macroalgae when all the sites were averaged together. There was also a trend of shifting from a drift algae-dominated site to a filamentous algae-dominated site. By the end of the study period, 5 out of 7 sites were dominated by filamentous algae. Polysiphonia was the most frequently collected macroalgae species during the entire study, and Hinxia was the 5th most collected species. In total, we were able to identify 49 different species of macroalgae in the Sanibel Bay area. So this is just a chart that shows the releases prior to the study period and during the study period. So during that summer, they had released a lot of water, but during the actual study period, the releases were basically within the recommended range. I think that the releases during the summer season had an overall negative effect during the study period of macroalgae diversity, and this temporal delay could be related to the distance away from the releases, which explains why Andy Rossi had such a high diversity in the beginning, because it was far away from the releases in the summer, but as time went on and the release reached their macroalgae, the dominant species, were able to grow more. But to really make that conclusion, you'd have to do a longer-term study. Okay, so here are the results for the seagrass. I grouped these 4 westernmost sites together because they had the most similar macroalgae or percentage coverage throughout the period, kind of. That's the gray part, is macroalgae percent coverage. And then this black line is for Thalassia percent coverage, and the dotted line is for shoot counts. I'm only going to show you the results for Thalassia because that's the only species of seagrass that's found at all those sites. At Andy Rossi, the seagrass coverage and macroalgae coverage sort of followed a similar pattern, but this was a little bit more dramatic and changed than this. At Wolfert Road, they kind of also followed the same pattern, but deviated a little bit in some other places. In Tarpon Bay, seagrass declined for the first 3 periods and then increased in periods 4, 5, and 6. And at Dixie Beach Boulevard, it also followed a very similar pattern too. But at Dixie Beach Boulevard, in period 6, there was a huge Lingbia event. And when we had collected all the macroalgae in a quadrat, there was up to 200 grams of dry weight per meter squared, which is a lot. There was also a Polysiphonia event during period 2, and there was up to 106 grams of dry weight per meter squared. One of the benefits of monitoring these sites on a monthly basis is being able to record these types of events. So these were the easternmost sites, and they also had similar macroalgae percent coverage changes, kind of. And at the Bailey Road site, there was a varying amount of macroalgae coverage, but seagrass remained consistently low. At the Causeway, there was a varying amount of macroalgae coverage, but seagrass continued to grow throughout the entire study period. And at the Toll Booth, seagrass was consistently high. So just by looking at these graphs, I couldn't really see any relationship between macroalgae and seagrass. Oh yeah, and there was a Lingbia event at these 2 sites as well. And here's just some pictures of Lingbia on seagrass. It's obviously very detrimental. It forms these sand, gas mats that can float around. They're really unsightly, and they kind of smell bad. So to get a better understanding of the relationship between seagrass and macroalgae, I just did this plot of percent coverage of Thalassia versus percent coverage of macroalgae. In my mind, I was thinking that this relationship would be negative, that as macroalgae increased, Thalassia would decrease. But there's actually a slight positive trend, even though it's not very strong. So I got to thinking and was looking at the graphs some more, and I noticed that sometimes when seagrass coverage was high, macroalgae coverage would be high too. And that's just because seagrass acts as a habitat for macroalgae. So it kind of makes sense that the more seagrass there is, there's going to be more macroalgae there. Over time, this could be detrimental to seagrass, but I wasn't really able to capture it in the short-term study. So therefore, you can sometimes expect more macroalgae in seagrass beds, especially in eutrophic waters. To see if water quality had anything to do with seagrass coverage, I just did a plot of Thalassia coverage versus CDOM and turbidity, which are measurements of stuff that's in the water, and I didn't see any relationship. Okay. So, unfortunately, with the small scope of the study, I wasn't able to determine the relationship between seagrass and macroalgae. This is probably because the relationship is extremely complicated and not very well understood. A longer, more detailed study with modified methods would probably be better suited to determine if macroalgae had a negative effect on seagrass in our estuaries. Other factors that contribute to seagrass decline include reduced water clarity from nutrients in the water and the development of shipping lanes and crop scarring. So I was still able to make a few observations from this study. When a macroalgae species becomes dominant, macroalgae diversity decreases, and freshwater releases can possibly have effects on macroalgae months after they occur. Increased seagrass density is related to increased macroalgae in nutrient-enriched water, and long-term trends can determine if macroalgae have detrimental effects. Monitoring these sites can help us determine when macroalgae blooms occur, what the species are, and can help us find patterns between nutrients and macroalgae composition. There's also a lot of further research that can be done based off of this research. To find out more about local macroalgae, we can do long-term monitoring of macroalgae at the same sites as here and in the Gulf of Mexico to track changes and document events. And to better understand seagrass and macroalgae relationship, we could redesign the methods to better answer the question. Maybe just do it in one location with many quadrats, have it be a long-term project, and collect more data on the water quality in seagrass. A lot of more questions can be answered through macroalgae physiology. So we could perform experiments on different types of species, such as a rare species, to see if, remember we were talking about the Km before, a rare species would have a really low Km, and we could sort of figure out connections between the rarity of a species and their physiology and how they react to nutrients. And we can also find connections between the composition of macroalgae and freshwater releases. Another cool thing to do with macroalgae to really complete the picture of what's going on with its physiology would be to determine the nutrient composition of the macroalgae after the experiments are done or during the experiments to really see what's going on with them. So thank you guys. Thank you everybody at the Marine Lab. You guys are great. Thank you Eric and AJ for helping me design my experiment. And also thank you to the volunteers for helping me sort through my macroalgae and also look at data and helping me out there. So does anyone have any questions? Mark?
Yeah, I was just wondering, would it make sense to look at phosphorus too if you're artificially increasing the nitrogen concentrations in your experiments?
Well,
is there a possibility that phosphorus might now be something that's limiting growth?
No, because phosphorus isn't really a limiting nutrient in our waters, so it shouldn't be limiting during the experiment, but it would. To really prove that, you probably should do an experiment to see if it's limited by phosphorus when you spike it with ammonia or nitrate. So yeah, that would be another good component.
Very good. Thank you.
Thank you.
Thanks for coming.